Welcome to Animal Biotechnology. Imagine trying to find a single spelling mistake in a massive library filled with millions of books. Traditional medicine often waits until that spelling mistake causes a book to fall apart before noticing there is a problem. Molecular diagnosis, however, is like having a highly advanced search engine that scans the text of the books directly to find the exact typo before any damage is done. Today, we will explore how we use this concept to diagnose genetic diseases. Now that we understand the basic premise, let us look at what molecular diagnosis actually entails. In biology, the typo we are looking for is a mutation in the DNA, and the library is the animal or human genome. By looking directly at the genetic code, we can identify diseases long before physical symptoms appear. This allows us to treat the root cause rather than just the symptoms. It shifts veterinary science and medicine from a reactive approach to a proactive and predictive one, which is absolutely critical for managing genetic diseases like sickle cell anemia or cystic fibrosis. This proactive approach brings us to our first specific technique, restriction fragment length polymorphism, or RFLP. Think of a long piece of ribbon that has specific markings on it where you are supposed to cut it with scissors. If someone accidentally spills ink over one of those markings, you will miss that cut, resulting in a longer piece of ribbon. RFLP works exactly like this. Our DNA has specific sequences where molecular scissors, called restriction enzymes, make cuts. If a genetic mutation changes the sequence at a cut site, the scissors will not cut there, leaving us with a longer piece of DNA. To understand how we track these cuts, we rely on a simple mathematical principle. The formula states that L total equals L1 plus L2 all the way to Ln. Conceptually, this means the sum of the lengths of the cut fragments must equal the total length of the original uncut DNA segment. L total represents the total length of the amplified DNA region in base pairs. The variables L1, L2, and Ln represent the lengths of the individual DNA fragments generated after the restriction enzyme cuts the DNA. If a mutation destroys a cut site, two smaller fragments combine into one larger fragment, but the total length remains perfectly constant. Before we move on, let us see this with a concrete example. We are looking at diagnosing sickle cell anemia using RFLP. We are given a normal beta globin gene region that is 1,150 base pairs long. It has 3 cut sites, producing fragments of 200, 500, and 450 base pairs. The sickle cell mutation destroys the cut site between the 200 and 500 base pair fragments. We are asked to calculate the fragment sizes for a normal individual, a carrier, and a diseased individual. We will use the fragment addition principle to solve this. Let us work through the mathematical reasoning for the mutant allele. Since the cut site is destroyed, the adjacent fragments merge into one larger fragment. We calculate the merged fragment size by adding the lengths of the 2 fragments that are no longer separated by the enzyme. L mutant equals L1 plus L2. We substitute the lengths of the 2 merging fragments, which are 200 and 500. Adding these gives us a new fragment size of 700 base pairs. The 450 base pair fragment remains completely unaffected because its cut site was not altered. So we have calculated the mutant fragment size. This brings us to interpreting the final physical results on a gel. A normal individual will show 3 bands, 200, 450, and 500 base pairs. A diseased individual with 2 mutant alleles will show 2 bands, 450 and 700 base pairs. A carrier will have both alleles, showing all 4 bands. This physical difference in band sizes allows doctors to perfectly diagnose the patient's genetic status. By simply adding up the lengths, we map out exactly where a mutation has occurred. Now that we have seen how RFLP works, we must address its limitation. Not all mutations destroy or create a restriction site. This leads us to the allele-specific oligonucleotide, or ASO, dot blot method. Imagine you have 2 identical-looking keys, but one has a microscopic scratch that makes it fail to open a lock. To figure out which is which, you create a highly sensitive mold that only perfectly fits the unscratched key. ASO works just like this mold. It uses a short, custom-made piece of DNA designed to stick perfectly to the normal gene, but it will fail to stick if there is even a single mutation. To make sure our ASO probe only sticks to a perfect match, we must carefully control the temperature. This formula, known as the Wallace rule, estimates the melting temperature at which half of the DNA probes will detach from the target DNA. Tm represents this melting temperature. The variables G, C, A, and T represent the number of guanine, cytosine, adenine, and thymine nucleotides in the probe. Because guanine and cytosine share 3 hydrogen bonds, making them stronger, they are multiplied by 4. Adenine and thymine share only 2 bonds and are multiplied by 2. If G or C increases, you need more heat to separate the strands. This formula we just saw leads us to an important practical application. A scientist designs an ASO probe to detect the delta F508 mutation in cystic fibrosis. We're given that the designed probe is 18 nucleotides long and contains 4 guanines, 4 cytosines, 6 adenines, and 4 thymines. We're asked to calculate the melting temperature to set the correct washing stringency for the dot blot. To ensure the probe only binds to a perfect match, the washing temperature must be set just below the melting temperature. We will use the Wallace rule to find this exact value. Let us apply the mathematical reasoning to find the melting temperature. We substitute the given nucleotide counts into the Wallace rule formula. G is 4, C is 4, A is 6, and T is 4. First, we add the counts inside the parentheses. The sum of G and C is 8, and the sum of A and T is 10. Multiplying these by their respective hydrogen bond factors gives us 32 plus 20. Adding these values gives us a final melting temperature of 52 degrees Celsius. This means at 52 degrees, the perfectly matched probe will start to detach. So we have proven that the theoretical melting temperature is 52 degrees Celsius. This brings us to the physical application of this result. To perform the ASO dot blot successfully, the scientist should wash the membrane at a stringent temperature just below this, around 50 degrees Celsius. At this temperature, a probe with even a single mismatch will fall off completely. It leaves a signal only if the patient has the exact genetic sequence the probe was designed for. This precise temperature control is what makes the assay so incredibly accurate. Now that we have calculated the exact conditions, let us look at what the actual test results look like. Here we see a schematic of an ASO dot blot assay. The diagram is divided into a grid. The rows represent different patients: a normal patient, a carrier, and a diseased patient. The columns represent the 2 different probes used, the normal probe and the mutant probe. Dark spots indicate positive hybridization, meaning the probe successfully stuck to the DNA. Notice how the normal patient only shows a dark spot under the normal probe, while the diseased patient only shows a spot under the mutant probe. The carrier shows dark spots under both, perfectly illustrating their genetic makeup. While ASO is powerful, it requires careful temperature control for every single probe. This brings us to another highly robust method, the PCR oligonucleotide ligation assay, or OLA. Imagine two pieces of a broken bridge that need to be welded together. The welder can only join them if the two pieces align perfectly, end to end, without even a millimeter of a gap. OLA uses this exact principle. It uses an enzyme called DNA ligase, which acts as the molecular welder. This enzyme will only join two pieces of DNA together if they match the patient's DNA perfectly at the exact spot where a disease mutation might occur. Because OLA relies on strict enzymatic precision, it is highly reliable for clinical screening. We measure this reliability using the diagnostic sensitivity formula. S equals TP divided by the sum of TP and FN, all multiplied by 100. Conceptually, this calculates the ability of the molecular test to correctly identify those who actually have the disease. S represents the sensitivity of the assay as a percentage. TP stands for true positives, which are patients with the mutation who correctly test positive. FN stands for false negatives, which are patients with the mutation who incorrectly test negative. If false negatives decrease, the overall sensitivity increases toward a perfect score. Before we conclude, let us see this formula in action with a clinical example. A new multiplex panel is designed to screen for 50 different cystic fibrosis mutations. We are given data from a clinical trial of 500 patients known to have cystic fibrosis mutations. The test correctly identified 485 patients as positive. However, it missed the mutation in 15 patients due to rare genetic variants. We are asked to calculate the diagnostic sensitivity of this panel. We will use the sensitivity formula, identifying the true positives and false negatives to evaluate the test's reliability. Let us work through the mathematical reasoning for the sensitivity. From the problem, the test correctly identified 485 patients, so TP is 485. The test missed 15 patients, so FN is 15. We substitute these values into the formula. First, we add the values in the denominator, which represents the total number of sick patients tested, giving us 500. Dividing 485 by 500 gives 0.97. Multiplying by 100 converts this into a final sensitivity of 97%. This proves the assay is highly reliable for high-throughput screening. We have covered a lot of ground today, so let us reinforce the most important ideas. First, molecular diagnosis allows us to proactively detect genetic diseases at the DNA level before symptoms appear. Second, techniques like RFLP use restriction enzymes to physically reveal mutations through changes in DNA fragment lengths. Third, when mutations do not affect cut sites, methods like ASO and OLA provide highly specific alternatives using custom probes and precise enzymes like DNA ligase. You now have a solid understanding of how these powerful biotechnological tools are used to save lives and manage genetic diseases.